Good morning, students. We are going to demonstrate the practical sessions through video recordings. Because of this pandemic situation, we cannot call you to the school and do the practical sessions personally. So we are planning to send the YouTube videos of practical sessions. We have only two practicals demonstration type, that is pollen germination in sugar solution and DNA extraction. Rest all spotters only. The spotters and its description and identifying features and all we will send you later through notes, and the microscopic figures also will be attached along with that. And based on this practical topics, objective related questions will be asked along with the theory or separate practical paper as instructed by the board. So you can confidently appear for that practical questions. Now let us come to the first topic, that is pollen germination in sugar solution. So to study the pollen, first we have to introduce what is pollen. So pollen grain or microspore is the first cell of the male gametophyte, and it is taking place within the anther. The formation, microsporogenesis, we'll say, it is taking place within the anther. And the pollen grain is uninucleate in the beginning, but at the time of liberation, it will become two-celled. That is a small generative cell and a large vegetative cell. So in this two-celled or even three-celled condition also, it can be liberated. So the microsporogenesis or the formation of pollen, it is taking place within the microsporangium. That means within the anther. Then the basic structure of pollen when we are studying, externally it looks, the shape of the pollen, the structure of the pollen will vary from species to species. So here we are going to demonstrate the pollen grain of hibiscus, as well as I am demonstrating the Vinca rosea, that means Shavanari in Malayalam will say, or periwinkle, that is a common name. So two flowers we are showing it, how to take the pollen grain and how to keep it in sugar solution and how to observe it under the microscope. So here the structure when we are saying the pollen grain consists of two layers. Outer layer, which is called as exine, and it is made up of sporopollenin, and an inner layer that is called as intine, it is made up of pectocellulose. And exine consists of 2 or 3 pores that is called as germ pores that will allow the germination of pollen tube. When the pollen grain is germinating on the stigma or in the sugar solution, the pollen tube emerges through the germ pore. Then the two cells already I said, inside a mechopollen, they contain two cells, a generative cell and a vegetative cell. Generative cell gives rise to two male gametes, and vegetative cell, which is vegetative in function and helps in formation of pollen tube. So this two male gametes, out of that two male gametes, one male gamete will fuse. When it reaches into the ovary, it will fuse with the egg that will form zygote formation that later develops into embryo. The other male gamete fuses with the pollen nuclei and they forms the PEN. PEN later develops into embryo sac. Not embryo sac, endosperm. So we'll move on to how we can prepare the experiment, or how can we make the slide of pollen germinated pollen grain. So first and foremost is you have to separate the pollen from the flower given. So it can be hibiscus, or even it can be vinca. So vinca, those who are not familiar, the figure I have attached here. So from the hibiscus, with the help of scissors, you can cut the stamen and you can dust it onto a slide which contain 10% sugar solution. So 10% sugar solution means 100 ml of water, and in that 10 gram of sugar, common sugar. You can add sucrose. You can add and stir it well. You can make a nutrient solution. To that, if boric acid is there, means a pinch of boric acid and all other necessary needed nutrients you can add. If it is not available, only sugar solution is needed. Then from that sugar solution, you can take a drop of sugar solution onto a clean slide, and to that you can dust off the pollen. So hibiscus, it is easy to separate the pollen. With the help of scissors, you can cut off the filament, and then you can add the pollen anthers into the sugar solution. And with the help of needle, you can twist it off. Twist it means you have to break that anther lobe. Then the pollens will come out. And you can, with the help of a cover slip, you can place the cover slip on top and wait for 30 minutes. Before that, if you want to see the pollen grain as such, you can keep it under the microscope and observe it through the 10x, and even for the further clarification 40x, you can observe it. Then wait for 30 minutes. So the first figure in this slide I have shown, immediately after separating the pollen grain of vinca. It is not the hibiscus, it is vinca. Hibiscus, the first previous slide I have shown, the exine is showing a spocky appearance. If you are observing the vinca pollen grain, it will be round, smooth surfaced only. The exine will be smooth surfaced and it will be intact. And after 30 minutes, that is the third figure which is given in this slide. After 30 minutes, you can see there are small extensions, small tubular extensions, which is coming out from some of this pollen grain, because slowly the sugar solution will be absorbed, and that will induce the development of the pollen tube. That means vegetative cell will come out, which will carry 2 male gametes, and that pollen tube will germinate, tube will form. That is called as the germination of pollen grain. So different stages of pollen grain, some of them will be germinated, some of them will not be germinated. So you can see and you can measure or you can count the number of pollens which are germinated out of the total number of pollens. So this is the small symbol experiment only. Now I am going to show the diagram, because always for the practical they will ask, describe the main characteristic features or comment on the pollen germination along with the diagram. So since we cannot represent the practical session, mostly the objective questions only will be there. So this is the external structure of a pollen, exine, intine, and a vegetative cell and a generative cell. The diagram, sometimes diagram based questions will be asked, so you have to label it. Vegetative cell will contain a distinct nucleus, irregular in appearance, and the generative cell is small and spindle shaped. It will be floating within the cytoplasm of the vegetative cell. And exine and intine, exine is made up of sporopollenin, intine is made up of pectocellulose. So that is the intact one. Then the B figure, it shows the early stage of pollen germination, which will carry the pollen tube will emerge out of the germ pore, and the pollen tube carry the pollen tube nucleus, that means vegetative nucleus, and the generative nucleus. Later the generative nucleus will divide into two and they forms the 2 male gametes. So that is the later stages of development. And the figure, sometimes the figure will be given and they will ask the questions related with that. And along with that, boric acid or boron is essential for the pollen development. And this is the figures, different stages of figures which we can observe through the microscope under 10x. So it is the stigma, in the pollinated pistil. This also I have included. So when the flower as such, when you are taking, and their pistil alone, if you are cutting and keeping it under the microscope, you can see there are different pollens are already there on the stigma, because as soon as the flower is open, from other plants, other flowers, the pollen will land on to the stigma, and the development of the pollen germination will be there, and at the same time the different stages of pollen tube development also can be seen through the microscope. So this is a microscopic view of germinated pollen grain on the stigma. The previous one I have shown is the development of pollen grain within the sugar solution. This one is on the stigma. With this, I concluded the demonstration part. Now I will show you the doing part, how we can observe, how we can separate the pollen grain, and how can we keep that one in sugar solution, how can we observe this one under the microscope. Now we will move on to the doing part. So first, the main materials which are required: cover slip, slide, flour, scissors, forceps, sugar, sucrose, and water. So approximately 10 gram of sugar in 100 ml we have to add and stir it well. Thoroughly we have to stir, and if you have boric acid, a pinch of boric acid can also be added to fasten the pollen germination, because it is the mineral which is required for fastening or for the essential for the germination of pollen. Now take the flower. With the help of scissors, you can remove the stamens. So you can take the scissor and cut the stamens and dust it onto the clean slide. So for this you need to extract, or you have to separate 1 or 2 stamens and place it into the center of the slide. Carefully have to do it, and with the help of needle you can tease the anther. Otherwise sometimes the anther lobe will not be open. So you can press it gently on the anther and pour 1 drop of sugar solution, which already we have prepared. Then take the cow slip with the help of needle or forceps. We can place it on top of the pollen grain. Then the base of the needle, or with a pencil or with your pen or with your finger itself, you slightly tap it onto the anther, so that anther will break and the pollen grains will come out. So gently you have to do, otherwise the slide cover slip will break. So you can just with the finger itself, you can press it gently on that and just squash it. So when you are squashing it, you have to be very, very careful, otherwise cow slip, it is very fragile only. So with the finger itself, like this what I showed, in that manner you can just press it so that you can see the pollen grains, that smashed appearance of the anther. So once that anther is completely smashed, then wait for approximately 30 minutes. But before that, it is well and good if you are observing this under the microscope, so you can see the pollen without germination. Okay, then after 30 minutes, you can see the difference between the germinated and non-germinated pollen. The same procedure itself I am here showing with the vinca flower, periwinkle we'll say, or Shavannari in Malayalam. So how can you extract the pollen from, because it is not visible externally. So first you have to remove, you have to pluck off that from the corollal tube and make a slit on the tube of the corolla. First petals, the external part of the petal, remove it, and with the help of plucker you can take that slit, open the corollal tube and take the stamen. It is very minute, 4 stamens will be there. From that you can, with the help of needle, you can separate it. So it is very minute only. With the help of that, you can remove it and place it on the slide. The same way what we have done with the hibiscus, same way pour 10% of sugar solution or sucrose solution, place it. There also you have to gently squeeze it or you can tear it off. That is to separate the pollens from the anther. Okay, so if you want one more, because since it is very minute, you can take one more anther lobe like this way and place it on the slide. It is very minute only, but even then very germination will be very fast or very easily can be detected in vinca. In hibiscus, the germination frequency is very, very slow and it will not germinate much. Whereas vinca means it is very easily it will germinate. So practical, when you are getting a chance to do in the lab, it is better to use vinca instead of hibiscus. But hibiscus, it is easily visible, very easily we can handle it. So whatever it is, either vinca or hibiscus, you can do it and press it gently, and after 30 minutes you can take this one and keep it under the microscope. Like this you can keep it. Then this is the figure which I have shown as the image of the ungerminated pollen in different stages. So you can see here X sign, that pollen germination in different stages. Small pollen tube which is coming out, then slowly it is enlarging, it is elongating, and finally you will get the image like this way. So this is only you have to do it, and this is the complete image. So thank you for watching.